It's my pleasure. It's my pleasure to introduce today Anya Brown. She got her BAs in Brown University in the year 2009, followed by a Master in Biology from California State University and a PhD in Ecology from the University of Georgia. And she performed her postdoctoral work in tropical ecology at the University of Florida. And right now, she's an assistant professor and a campus scholar here at UC Davis. She's located in the Bodega Marine Lab. And today, her research focus mainly in studying community ecology, particularly in coral reefs. Very interesting topic. And today she's talking to us about the ecology of extended phenotypes, stressor effects on coral hosts and their microbes. So welcome, Anya. Happy to have you as a campus scholar, and please go ahead.
Thank you so much. I'm really excited to tell you a bit about my work. Like Veronica just said, I'm new to Davis, and I'm based at the Marine Lab, and so it's just great to talk to everyone beyond the group that I normally interact with. I'm going to talk a little bit more about my academic journey, which, as I understand, is part of the talks for the campus colloquium. So I started my degree at my undergraduate degree at Brown University, and one of the formative experiences that I had while I was there was as part of the 3Cs program, where I was lucky enough to visit 3 different marine stations located in different areas of the world, including the Northeastern Marine Science Center, one in French Polynesia, the UC Berkeley Gump Marine Station, and one in Southern California, the USC Wrigley Marine Station. And this is where I started to fall really in love with the field stations and had these transformative experiences that have helped mold me into the scientist that I am today. As part of my experience with 3Cs is I met my master's advisor, who was a professor at Cal State Northridge, where I did my master's degree, and continued to work in French Polynesia at the Gump Marine Station. And that's where I began some of my interest in corals and coral microbial interactions, which I continued during my PhD. I started my PhD at the University of Florida, still working at the same field locations. But my advisor moved 2 years into my degree program, and so I moved as well. And so I finished at the University of Georgia at the Odom School, where my PhD is from. I popped back to UF for a 2-year postdoc, where I started working with NGOs an NGO in the Caribbean, the Central Caribbean Marine Institute, which I'll talk a little bit more about some of that work today. And then I did my last postdoc at the Woods Hole Oceanographic Institution, where I started working on temperate corals. And now I'm at UC Davis and based at the Bodega Marine Lab, which is on Horseshoe Cove. And you can see the building right here in the background, which is a really beautiful place to work, even though right now there's it's a bit cold. And the way that I've been liking to start a lot of my talks is with a question. And that is, when I say phenotypes, what comes to mind? Maybe it's morphological traits like body size, or physiological traits like growth rate, or behavioral traits like hiding from predators. But what about the extended phenotype? And the extended phenotype is the effects of a gene that go beyond an individual, which was first conceptualized by Dawkins in 1976. And an example of this that I really like are between these 2 sister taxa of mice, one that builds a short, simple burrow, and one that builds a long multi-tunnel burrow. It's these burrow characteristics, they're an example of the extended phenotype, as it influences how these mice can escape from their predators, affecting their fitness. Another example that I really like of the extended phenotype is between host and parasites, where the host is the extended phenotype of the parasite. So for example, with these frogs, when they're infected by the trematode parasite, they grow extra limbs. And so that makes them swim slower and more easily captured by the parasite's next host, in this case, birds. And more recently, the concept of the extended phenotype has been extended further to include the host microbial partners. And so host-associated microbiomes, like these associated with host tree roots, can be host-specific, and they can lead to changes in the environment or to the host that can influence fitness. And so like burrow size in mice and host characteristics of parasites, we can think of the microbiome as part of the host's extended phenotype and a trait of the host. And so in doing so, we can think about how temperature, salinity, predators, these abiotic and biotic environmental characteristics can affect the microbiome, can affect the host, or can affect both, leading to a new phenotype or a plastic shift in a phenotype. This might help species expand into new areas or quickly acclimatize to changing conditions. It could also have deleterious effects when stressors break down the associations between these symbionts and their hosts. And I like to think about microbes as part of the host extended phenotype, in part because it allows us to think in terms of evolution, as changes in the host microbial community may lead to adaptive benefits or disadvantages to the host. In terms of community ecology, as species interactions can be modulated by changes in the microbiome, as well as microbial ecology, as the dynamics of the microbiome are influenced by the environment, other microbes, and the host itself. And a question that really motivates my work is what the implications are for variation in traits. And when I say traits, I include microbes. And my research and my lab, as it's developing, is really focusing on these four concepts: the stressor effects on organisms and populations, or how variation in species responses to stressors can be influenced from the microbial to population scales; microbially mediated species interactions, or incorporating our understanding of microbes into our understanding of classic species interactions, like competition; adaptive plasticity, or how previous exposure environments can modulate future species responses and whether it's mediated by microbes; and community phenotypes. So thinking about the indirect community-wide implications mediated by variation in species traits. And today I'm going to hit on three of these concept areas and focus on, this take-home point that I'm already sharing with you, that's trait variation and response to stress has wide implications for populations and communities. I'm going to provide two examples, one that focuses on a stressor, a vermetid snail, and one that focuses on a different stressor, a disease. And then I'll talk a little bit about my current and future work while here at Davis. And really the star of the show today is going to be corals. And I just want to make sure we're all on the same page on what a coral is. So here's a photo of a massive Porites colony. And corals, when you're looking at them and you see that mustard color in the case of this one, you're seeing the color of their tissue layer. And the tissue layer is a thin layer that's covering the skeleton and producing that skeleton. So when you see a coral, you're actually seeing years and years of skeletal deposition, and the living portion is just that thin layer on the top. And that tissue is composed of interconnected polyps making up that coral colony. And if we were to zoom into one of those polyps, it looks something like this cartoon. Those tentacles capture food in the water column, allowing the coral to feed heterotrophically. Although most corals, if you look on their insides, you'll find their algal symbionts. A common name for them is zooxanthellae, and they're in the family Symbiodinaceae. It's these algal symbionts that are responsible for a large portion of the coral's nutrition as they transfer photosynthate to the coral host. Corals also have a surface mucus layer. So not that I'm recommending touching a coral, but if you were, they feel pretty mucousy. And in that surface mucus layer is a rich microbial community that includes bacteria, fungi, and archaea responsible for nutrient cycling in the host, as well as pathogen defense. And for the kinds of questions that I'm interested in studying, corals really do show fantastic amounts of variation, making them an excellent study system for thinking about variation within species and among species. And that variation can include differences in growth or morphology. It also includes variation in their symbionts, including the types of algal symbionts, as well as their densities, as well as their microbiome and that surface mucus layer in particular, because it's really the first part of the coral that's in contact with the environment, and the first line of attack against potential incoming pathogens or stressor microbes. And disruption of these communities can be good indicators of stress. And it's these corals that are the foundation of the ecosystems that are characteristic of tropical environments. And this picture is just really exemplifying in some ways the diversity of the different types of corals that are there and that provide really important functional roles for this system. However, corals globally are threatened by a multitude of stressors. And one of those stressors is this vermetid, Ceraesignum maximum. And vermetids are snails, although they look a bit more like worms, and they're super unusual. You probably are imagining a snail like you would see on the coast of California that's more like has a coiled shell and I don't know, looks like Turbo the snail moving across the land or the sea. But these snails, like I said, are unusual. They're embedded into the reef matrix. So they are only mobile in their larval phase. They settle onto the reef, embed themselves into the reef, and so they don't move after that point. I'm pointing to 5 of them right here, just to give you a search image to what these look like. I think they kind of look like the operculum looks like cyclops eyeballs. And so another thing you might notice about this, and is a huge characteristic of these large worm snails, are these mucus nets. And it's these nets that are how they're able to feed. So like a spider's web, they put it over the surface of the coral or over the surface that they're in contact with, and it collects particles from the water column that the snail then consumes the net and all of its contents. And it's this mucus net that's been implicated in negative effects on coral. And that includes lower coral cover, reductions in coral growth, lowered coral survival, lower photosynthetic yield, as well as lowered water flow at coral surfaces. And what this means effectively is it's suffocating corals when it's present by decreasing oxygen because it's lowered water flow, not allowing for the exchange of oxygen with the overlying water column. It also changes coral morphologies from more rugose or bumpy structures to flatter structures. It has community-wide implications as they can decrease herbivory on these small algal turfs that you can see in this image behind here, which look like a fuzzy substrate. And these measures of negative effects were really strong, or had really strong negative effects when they were first observed in Moorea, French Polynesia in the early 2000s, particularly the growth effect. This arrow strength, this is an indicator of, the boldness is an indicator of the strength of that vermetid effect. However, subsequent studies and years show little to no effect of vermetids on corals, particularly on their growth. And so why would there be this lack or this missing effect? What happened to the strength of the effect on vermetids? Well, over this time period, several changes happened in the back reef of Moorea, French Polynesia, where these studies were conducted. There was a huge increase in the density of vermetids, nearly doubling in the time period where the initial studies happened, which is this area right here, and when the later studies occurred. And we know, as I told you earlier, that there's many different ways in which corals can vary, but they're also highly plastic. And that means they can change in response to environmental conditions. And they have microbial communities that are also shifting in response to environmental conditions. And so I asked if prior exposure to vermetids, now that they're in contact with them more in this later time periods, could that affect their response? Could that affect the coral's response to the snail? And this is a concept, or a way to think about this is, does previous exposure to vermetids decrease their effect on corals? And there's a concept in ecology, evolutionary ecology, called adaptive plasticity that this reminds me of. And I really like this example of it from Ald and Relia, 2011. And so here we have Physa acuta freshwater snails that have either been exposed to predators or have not been exposed to predators. So the larger body size are ones that have been previously exposed to their crayfish predator. Upon a secondary exposure, we see a reduction in the effect. So fewer or more survived, or more snails survived if they've been previously exposed to predators. Whereas the snails that have never been exposed to predators, we see a large effect, so more died. And on reefs, we have some corals where vermetids are absent. So there are none of these worm snails present on them, and some where vermetids are present. So they have them. So there's different exposure histories. And because of these differences in exposure history, I expect with response to secondary exposure, a large effect on those corals that have never been in contact with vermetids. And so that could look something like this, where a trait like growth rate on the y-axis would show a large deleterious effect when placed on a vermetid present reef, compared to corals that have been previously exposed to vermetids, where I expect to see a small effect. So once placed on a vermetid present reef, I don't expect to see a large difference compared to when they were on a vermetid absent reef. And so to test this hypothesis, I conducted a reciprocal transplant experiment. So I took corals from one of the previous exposure environments, indicated by orange for vermetid absent and purple for vermetid present, and transplanted them onto a reef, either with vermetids or without vermetids. And then the traits that I measured were calcification, a measure of coral growth, tissue thickness, which is about allocation of energy, where thicker tissues indicate more resources available, symbiogenesis density, another indicator of resource availability, as well as an indicator of the one of the symbionts, variation in the symbionts, and microbial communities, where I'm going to be looking at differences in diversity and shifting composition as indicators, potential indicators of stress. And this is what the corals looked like when they were planted out into the field. So here's one of the small coral fragments, originally from another reef, placed onto either a reef with or without vermetids. And so for the results of this, I'm going to go through this first plot slowly, where here we have transplant reef on the x-axis and change in skeletal mass on the y. Here, the colored of the circle, inside circle, indicates the previous exposure environment, and the outside circle represents the transplant environment. So in this case, corals from vermetid absent reefs are transplanted back onto vermetid absent reefs. When those corals originally from vermetid absent reefs are transplanted to vermetid present reefs, we see a reduction in their growth. And this is the same type of effect that was seen in those early studies. So now corals from vermetid present reefs, when transplanted back onto vermetid present reefs, show actually higher growth, but they show even higher growth when transplanted to vermetid absent reefs. So here we show an effect of the previous exposure environment, as well as an effect of the transplant environment. However, in general, the presence of vermetids leads to decreased growth, no matter what the origin. And we see several traits that show plasticity in addition to calcification. And those are microbial traits in terms of alpha diversity, where alpha diversity increases when corals are transplanted to vermetid present reefs and shifted depending on transplant environment. So shifted towards, in this NMDS plot, the vermetid present effect is shifted towards the NMDS2 axis. All of these traits, though, are pretty indicative of stressful conditions for the coral. And in the microbial composition shifts, that's in particular because the groups that are changing are associated with anaerobic conditions, indicating that there's likely, like I said before, at the surface of those corals, when the nets are present, there's a drawdown of oxygen, allowing for groups associated with low oxygen conditions to proliferate and groups associated with aerobic conditions to decrease. So these, again, are really indicators of stress more so than adaptive plasticity or helping the coral adjust to the presence of vermetids. Interestingly, though, we saw some traits that did not show plasticity, and that is both symbiogenesis density as well as tissue thickness, which were always higher on corals originally from reefs without vermetids. And in fact, we saw several traits that show some effect of that previous exposure environment. So why does previous exposure or the history of the coral really matter? Well, I've been really suggesting that vermetids are driving the effects or the phenotypic effects of the corals. But corals are highly plastic and show variation within and among species. And so sometimes it's even difficult to distinguish what corals are different species. So instead of what phenotypes or what do vermetids do to coral phenotypes, maybe I should have been asking what types of corals have vermetids? Or another way to think about this is there genetic variation among corals? And so to test this, I sequenced the mitochondrial cytochrome the MTCO1 region, and found, yes, there's consistent genetic differences between reefs. Corals where vermetids are absent or consistently genetically different from corals that are vermetid present. And that's what this tree is showing, as the corals that had vermetids are grouped together here. And there's a 0.4% sequence difference. And what that means is that there were 3 consistent places on a 700 base pair fragment that showed differences in these 2 corals. And so what this means is that the presence of vermetids depends on genotype. And really the vermetids are telling us the genotype of these corals much better than we were able to identify it by eye. But because of this finding that the presence of vermetids depends on genotype, it reminds me of this concept I brought up at the beginning, this idea of the extended phenotype and the effect of a gene beyond the individual. And Thomas Whittam expanded this idea into thinking about community phenotypes, or the consequence of extended phenotypes. And in the example that he gives is between Pinus edulis, the dominant member of the pinyon juniper systems, and these spongy moths. Different trees show differential susceptibility to attack by these moths. Some are susceptible and some are resistant. Thus, the moth is part of the extended phenotype of the tree and can have these community consequences, including destruction of shoots, turning trees into shrubs. This different susceptibility to moss can influence seed dispersal and fungal communities. And so the extended phenotype of these trees can have really wide-reaching effects for their community that are potentially heritable. And this is really analogous to the coral system, in part because Massoparietes are dominant members of this ecosystem. And I've just showed you that vermetids are genotypic, are influenced by different genotypes of the corals, suggesting that they're part of the extended phenotype of the coral. And so what this could mean, if we were to play this out over time, is we have a resistant and a susceptible to colonization coral. Resistant corals, if there's a partial mortality event, you'd expect to see a faster healing time because they have resources through their higher symbiogenesis density and thicker tissues, indicating there's more resources available for healing, leading to a coral that's intact. Susceptible to colonization corals, with their lower symbiogenesis densities and their thinner tissues, we expect to see slower healing, allowing for those vermetids to settle to dead spaces on those reefs and proliferate, because we know that they promote lower herbivory. We expect to see higher algal cover and more diverse microbiomes, degrading those reef environments. So depending on the presence of the resistant and susceptible genotypes, we expect potentially different reef trajectories. When the resistant to vermetid colonies are present, it would promote higher coral cover. And when the susceptible to snail colonization reefs are higher in relative abundance, we would expect reefs with lower coral cover. And so the different underlying genotypes leading to these cryptic differences in the dominant species on the reef can lead then to the development of these different types of communities that are potentially heritable. And this concept has been considered in terrestrial ecosystems, but is only now really being thought of as of the role that it might be playing in tropical coral reefs. And I think has really important implications in seagrasses as well as corals, and might help explain some of the variation that we see in communities. It also has implications for conservation, as the genotypes that are being planted on the reef can have much more far-reaching consequences than we previously thought. However, Vermetids are really not the only stressor on reefs. And in fact, they can be pretty localized compared to some of the other more global effects that some of the stressors are having on corals. And one of the stressors that comes to mind is coral disease. And this is a map just showing different tropical regions and the number of diseases attributed to those regions. And you can see in some of these places, there are really hot spots, so really red points suggesting hot spots of disease. And one of those areas is the Caribbean, which has been hard hit by disease on multiple different fronts. But what I really think of as the poster child for disease in corals is white band disease. And here's an Acropora cervicornis or staghorn coral affected by white band disease. For white band, the disease starts towards the middle of the colony and then extends outwards. And you can see that's happening here a little bit in the arrow that I'm pointing at. And as a close-up at the disease lesion, you can see active sloughing off of the brown coral tissue, leaving behind a dead white skeleton. And we know some things about this disease that first became prevalent in the 1980s. It's infectious. It transfers via currents, consumers of corals, as well as direct contact between colonies. There are several putative pathogens associated with the disease, including Vibrio harvii and Rickettsia-like organisms. But over from the 80s to the 90s, they led to 80 to 98% population losses, really changing the landscape of the reefs in the Caribbean in particular, as these corals were once the dominant coral on reefs. But it's in response to that there's been an influx in coral nurseries, which act as tree nurseries as they try to grow corals and then plant them back onto coral depauperate reefs or coral depauperate areas. And so this is a PVC frame from the Central Caribbean Marine Institute's ocean-based nursery. And on it, you see multiple colonies or multiple colonies being grown on that frame. And here's just an image of the nursery from going down into it, where you see multiple frames. And the way that coral nurseries are typically built, and definitely the way the CCMI nursery has been composed or was created, was first finding donor colonies from around the reef. So sourcing those the colonies on the frames from these donor colonies that are different genotypes. And here is just an image of little caiman, and each of these small colored fragments indicate a different genotype of corals. And once they're brought into the ocean-based nursery, in this case, they start to be placed on these frames in different fragments, and then they're really growing in this common garden environment, allowing us to remove any environmental effects of their original environment and understanding what the genotypic differences among them are. And so we know that when they're grown in this common garden environment, genotype has an effect on coral growth, and it also has an effect on their microbiome. So this PCA plot shows that the yellow genotype in this case differs from the green and red genotype. However, if we were to look a little bit closer of the microbial communities associated with these different genotypes, these different blocks indicate the 3 different genotypes. We see that all of them are dominated by this one member that's in light purple. And this is the genus MD355 or Candidatus aquarickettsia. So regardless of genotype, all of these corals are really dominated by this one genus in particular. And so although there are a lot of benefits to growing in the nursery for the corals, that they're not affected by some of the stressors that corals experience on reefs, like competition with algae, they still experience some of the widespread effects that stressors of coral reefs have, including disease like white band disease. And that's what happened in the CCMI nursery while I was working there. But it allowed us the opportunity to ask questions that we wouldn't normally be able to ask in wild environments, including what factors are influencing the disease spread in this semi-closed population, and what are the microbial shifts associated with disease. And in this case, we first noticed the disease in early May and almost immediately began sampling the disease over a 6-month period until we started to see the increase in a secondary disease in September. And what this means is that corals on each frame were visually monitored weekly and designated as either healthy, diseased, or dead. And when I say they were visually monitored, I mean 650 something corals were examined by a team of people led by the NGO research restoration manager, Dagny, and some of the key students who worked with us on this project. This was really a huge group effort. And this is what the data look like. So the orange color here indicates a healthy coral. The gray data is the diseased colonies, so where there was any incidence of diseased, and the black are dead colonies, so where there is no live tissue present at all. And we see that the disease peaked in around mid-July, and we actually did see some recovery of colonies that I'm not going to talk about today, but that was a nice, an interesting find of the study as well. And here's just an example of what the corals looked like on the frame, with some dead colonies, some live colonies, and some partially dead colonies, or some of the diseased colonies. And because there are multiple frames that make up this nursery, we can actually look at this data frame by frame. And I know this is a lot, but this is an example of what that looks like, again, with the orange indicating healthy, the gray, the diseased, and the black, the dead. And when I first saw this, a pattern really jumped out to me, and maybe you're noticing it too, but there are several of these frames that show more healthy colonies than diseased or dead, or really no disease, not that heavy of a disease pattern over time. And that's because there's something that I haven't yet told you. Oops, I just noticed my OneDrive notification. There's something that I have not told you about these frames, and that there's differences in genotype mixtures on the frames. Some frames have mixed genotypes and some frames have single genotypes. So when they have mixed genotypes, that means that there are colonies from multiple original donor colonies. And when it's single genotypes, there are multiple colonies on the frame, but they're all the same genotype. And in disease ecology, there's a concept called the dilution effect. And in brief, the idea behind this is in mixed groups, whether it's mixed species assemblages or in this case, mixed genotypic groups, we expect to see lower prevalence of disease. And that's because there's likely members of that mixed group that are resistant to disease that prevent transmission from one susceptible to another susceptible individual. On the other hand, a single species or a single genotype group, we expect to see higher prevalence because they're all shown, you would expect for there to be they would all be susceptible. So to test this with these data, I separated the frames into single genotype or mixed genotype frames to look at disease prevalence. And when doing so, what we see is corals on single genotype frames have higher disease prevalence compared to corals on mixed genotype frames. So at a population level, mixed frames show disease resistance. So what might be driving this effect? Well, implicit in this hypothesis is that there's differences in prevalence, or there's differences in susceptibility to disease depending on genotype. And so we asked that on the corals that either had were present on both single and mixed genotype frames. And so here's an example of 3 of those genotypes, and what we found were some genotypes were susceptible. So they were susceptible to disease when on single genotype frames, but show resistance on mixed genotype frames. So by themselves, they're susceptible to disease. Whereas the G genotype was resistant. So it shows low disease prevalence no matter what frame it's on. So what this suggests is that differences in disease susceptibility are really likely driving the population level disease resistance. And I think it's really obvious when looking at these 2 frames. One that has mixed genotypes that include the green or the resistant genotype, and one that's all susceptible genotypes. When the resistant genotype is present, no disease. When the susceptible genotypes are present, we see disease and death. So in addition to looking at these population level effects, we wanted to understand the microbial shifts associated with the disease. And so to look at that, we took mucus samples from healthy colonies that didn't show any sign of disease, and then mucus samples from diseased colonies in 2 locations, at the diseased lesion, shown in gray, and apparently healthy tissue, shown in this darker brown, which was 10 centimeters or more away from that diseased lesion. And here I'm going to show you the relative abundance of different genera of bacteria that were significantly different depending on treatment type. And this is based on 16S, the 16S rRNA gene, or so the bacterial communities. And so here we see several taxa that are disease associated. So the higher relative abundance in disease colonies or in disease samples compared to in the apparently healthy or healthy colonies. Including in this is this Vibrio that is the same species of Vibrio that's been previously associated with white band disease. But I really want to focus today on this Candidatus aqua, the one that's associated with healthy corals and shows higher relative abundances in both apparently healthy and healthy tissues. And this is an interesting ASV, or this is an interesting taxon rather. It's been consistently associated with healthy tissues, although in this study, but in previous work, it's been associated with disease and stress, and high abundances are associated with susceptibility to white band disease. But we can test that second hypothesis because we know the susceptibility of the genotypes in this study, where green or G is resistant and then the other 2 are susceptible. But there's not really any evident or clear evidence of differences in abundance with this genus when we look at this kind of coarse level of understanding of differences in relative abundance. But this is, again, looking at the genus level. If we were to look at more of the strain level, so base pair differences in this genus, we see something interesting. So here we have the resistant genotype, and each of these smaller bars indicate different strains of Candidatus aqua, and then the 2 susceptible genotypes. And again, each of those colors indicate the colors of those colored bars indicate the different strains of Candidatus aqua. And so what this means is that the different strains of Candidatus aqua are associated with colony disease resistance, because only ASV1 is associated with the resistant genotype. And so we're looking from the population to the microbial scale in this study. At the population level, we see this emergent effect based on a dilution effect that mixtures of genotypes increase disease resistance, which I'm suggesting is modulated by variation in genotype resistance at the colony level, and is associated with differences in the strain of dominant taxa associated with disease. These dominant taxa are associated with differences in disease resistance and lead to some really nice hypotheses about future work on what that might mean. And so to wrap up, I showed 2 examples of how intraspecific or cryptic species influences traits in response to stressors and how that might affect community and population responses. In the first example, depending on the resistance or susceptibility to vermetid colonization, there's the potential for different reef trajectories, one that's dominated by corals or one that's not dominated by corals. In the coral nursery, we saw differences in population level disease modulated by intraspecific differences in colony resistance or susceptibility to disease, which may be associated with microbial communities. And a big question that I'm continuously excited to think about is the interplay between host and microbial variability. And one way that I'm working on trying to understand this is this type of response to stress is with temperate corals, while I was at the Woods Hole Oceanographic Institution working with dormancy. And so corals, these temperate corals of Strangia poculata undergo dormant go dormant in the winter. So they're triggered somehow, somewhat temperature related, into stopping to halting their physiological responses in the wintertime, looking like this one image that says during. So they pull their tentacles in. They don't respond to any tactile stimulation. And it's not until temperatures start to rise do they start to emerge, and you can see their tentacles and they start actively feeding again. Some of the work that I've done previously while I was in Woods Hole suggests that this shift from before, during, and after dormancy in the host, it corresponds with microbial turnover, particularly a loss of copiotrophic or nutrient-loving microbes as corals enter dormancy, and an increase in microbes associated with nitrogen cycling and production of chemical defenses during dormancy, suggesting that there's some potential active removal of microbes as well as an active replacement of nutrition by the coral host. And so this is an active direction as well in the future new lab, or my current lab, which is based, again, as out at Davis, or out at the Bodega Marine Lab. And I'm really excited to continue thinking about these different concepts, both in coral reefs, temperate and tropical, and expanding into the seagrass habitats really right outside the front door at BML. And the projects that are really starting to take shape right now are with seagrass wasting disease. And so, like, corals are affected by diseases, seagrasses are too. In this case, we have a known pathogen. It's a slime mold, Labyrinthella zostera, which affects seagrass leaves. You can see the lesion right here, including, and here's another image of them taken by Karolina Zabinski, who's a graduate student of Jay Stakowicz and who's working on these types of questions as well. And so with my new graduate student, Serena, who's co-advised with Jay Stakowicz, we're going to be looking at variation in the pathogen in different areas of Tamales Bay, the neighboring bay from Bodega, and specifically trying to understand how the pathogen response is influenced by their natal environment and what role temperature or thermal adaptation has played in that. So stay tuned for the future work done by my new lab. And I want to thank you all for listening, and thank my collaborators on these different projects, my field and lab support, as well as all of the undergraduate, both field and lab interns who've helped make these projects a reality. And with that, I'll take any questions. And again, thanks for listening.
Thank you so much, Anya. So do we have any questions? So, Anya, I want to ask you. So I didn't understand very well how do you culture your algae? Are they in the sea inside? It's a silly question. Or do you have them in the lab, in a tank, or how that works?
So the algae, like the ones that...
The experiment that you did with the frames, mixing the species.
This one, the coral.
Yes.
On the different
that
frames.
one. That one.
Those were all ocean based. So the frames looked like this, and everything was in the field.
So where do you get the coral from? From the area?
Yeah, so that was from all of these corals this study happened in Little Cayman, in the Cayman Islands. And so the corals originally came from different locations around the island, and then had been growing in this coral nursery for several years before or different parts of the coral have been growing in the coral nursery for several years before I started working on them.
Okay, so I guess your students should be good divers, right?
Yeah. The nursery was at about at 60 feet, so there was a lot of diving that went into this, and again, a big role was played by the lead restoration manager. her assistant, and then a team of volunteers that like she was able to wrangle to help do some of these surveys.
Okay, we have questions. Lydia, please. Can you unmute yourself?
Yep. Hi.
Hi.
So very, very interesting talk. Congratulations. I think it's great that you also talk about your trajectory in in this progress. So I had a question. I had two. So it seems like there might be some type of coevolution to resist these type of invasives or these type of organisms that sort of suffocate the coral. And then you could talk about, you talked about how those that were resistant to disease showed a genetic difference. Is there anything particular about those places on the genome that showed that resistance? Like, are they related with a particular protein or up or down regulation of other genes? Like, is there any exploration in the transcriptomics? And then if you could expand how other stressors might modulate these interactions, because down here we're looking at how seaweeds might have some of the same responses and how different they are in Mexico, where they're exposed to thermal stress, compared to in California, where they're in much colder waters. So wonderful talk, and thank you for taking my question.
Yeah, okay, I'm going to start with the first one. I think that it does seem as though there's some coevolution between the corals and the vermetids, and that was I think that's an open question and one that I'm hoping a future student might be interested in delving into further. Question 2 was about on the genome of the corals that are resistant and susceptible, if there's particular regions that I know of that would help differentiate why they might be susceptible or resistant. I don't have an answer to that. The only reason why I know that they're different is because of some previous work that had been conducted that was mostly looking at population genetics within the region, and used corals from this nursery to show differences, population genetic differences. And so that, I think, is another area that's really ripe for study. Now that we know these phenotypic traits of these corals, what is it in the genome that might give us some answers as to why that might be? And I'm hoping to think about that both, or future studies could look at that both in terms of the host and as well as in the microbes. And then their third question is thinking about other stressors, and would we likely see similar responses, for example, with algae or with seaweeds. So that's actually when I originally went down to work in the Cayman Islands, I wanted to look at coral algal competition and how that varied with genotype and site. So I don't know yet, but I think that would be a really nice way to try to get an idea of the consistency in some of these stress responses. In terms of the microbial response to seaweeds, that can be have similar general effects as vermetids do. So when seaweeds are exposed to corals, you tend to see increases in alpha diversity and more dispersion, so more spread of the microbial communities. There was just a talk by a postdoc here who's visiting and is interviewing for a postdoc physician, who's also showed that the amount of seaweed on a reef can also affect coral microbial communities and the response of coral microbiomes to seaweed. So that is, I think, a somewhat more complex question that I'd be happy to talk with you further about, because it's something I'm also really interested in.
Cool. Thank you so much. Wonderful talk.
Thanks. I see Kenzie's hand is up.
Hi, Anya.
Kenzie?
Can you hear me?
Yes.
Okay. Wonderful talk. It's all very, very exciting. I was wondering, since you found higher diversity, or that it's known that there's higher diversity associated with higher resistance, if within a microbial community, if there was a higher diversity of like the Symbiodinaceae within the coral, if that correlated with a higher resistance, or if it's more dependent on the dominant strain that was present within that microbial community.
Yeah, so in the case of the disease, and I appreciate that I gave two examples with disease with susceptible and resistant and showed different types of results. So the increase in diversity of the microbes was with the vermetid coral example. And I didn't show differences in diversity with the disease example per se. For those, the disease signal kind of swamped out a bit of the genetic the genotypic differences, and so I didn't show that data, although I'm still working on some parsing that out a little bit, because the coverage of samples for the disease was a little bit low on the up for different genotypes in terms of the microbial data that worked. But I think your other part of that question is there an expectation for variation in the algal symbiont identity and susceptibility or resistance to disease? And I don't know if I don't know the answer to that right now, but I think that's an interesting kind of question also about thinking about symbiosis, and like if you expect a tight symbiotic interaction to be more or less resilient to disease versus a like more generalist symbiotic relationship to be more or less susceptible to disease. So I think there's some interesting ways to think about that.
Yeah, definitely. Thank you.
More questions? Do we have more questions?
Looks like Carolina has a question.
Carolina?
Hey, Anya, great talk. Okay. I was thinking about this slide that we're on and how you found that the resistant genotypes had like certain strains of this one genus of bacteria. Like when I'm starting to think about symbioses and incorporating the microbes with that, I think of the microbiome as like the genetic repertoire as the extension, like the functions of these microbes. So I think I might have missed this, but did you sample for the microbiome like after a period of time, or did you notice that these resistant genotypes like had an influence on the microbiomes of the susceptible ones, where like they got that certain strain of microbe after a certain time, or if it's, does that make sense? Or
yeah, and I would say stay tuned. So the microbe data I showed was a combination of at the initial time point of the disease and from a year prior, where it was really just looking at healthy corals and the makeup of the healthy coral, or the microbial community, particularly the different strains of that one genus in healthy colonies that had not yet been affected by the disease. And so the data that is currently in the works right now is looking at the changes in the microbiome over the course of the disease. And I should have said that was when I had talked about that part of the sampling design, is that data is not what I was showing today. It was just the initial time point for during the disease, and then the other data was from earlier. But I think that's a good hypothesis in terms of when corals recover, are they also gaining or changing in the relative abundance of this putative bacteria that might be influencing resistance or susceptibility. And I would say that part of this is still in the hypothesis realm. It's just an association at this point.
Okay, cool. Super cool. Thank you.
Yeah.
Mariel? You are muted.
Yes, thank you, Anya. This was a great talk. I have a question, and I was thinking before you talked about the white band disease, you talked about the vermetids, and you showed that there's 0.4% sequence difference between corals susceptible to the worms and those that aren't, leading you to conclude that the presence of vermetids depends on the genotype. And before you started talking about white band disease, I was wondering if you could do the experiment repeated in the control conditions. So with a specific, focusing on a coral with a specific genotype. Is that possible, or is this not possible in this particular case?
Do you mean?
So just looking at corals that are susceptible and exposing them to the worms or not, and comparing that to a population of corals that are not susceptible. Because here your populations seem to be mixed, and you were able to distinguish them when you did the genotyping, but you couldn't really distinguish them by looking at them, right?
So so I wasn't able to distinguish them until I did the genotype, but I still used so these corals that I did the genotyping with are the same corals that were in the experiment that I described. So these are the same ones that that the data that I showed you before this are based on. So it was more a priori the original. So that, like, if I were to re-show this, like these results, for example, another way to think about that is that inner circle represents the genotype without vermetids. So let's call it genotype A. And then the purple inner circle represents the genotype that allows for vermetid colonization. So the colonies that were the genotypes that were not susceptible for vermetids to vermetid colonization are present in this were present in the experiment, and the colonies that were susceptible to vermetid colonization.
You knew a priori that they were different.
Well, I mean, I did not know a priori, but because of the strong signal, it happens to be that the way that I set up my experiment, also you could interpret it in both ways.
Okay, that's very interesting. Really interesting. Thank you.
Any more questions?
Veronica, there's a question in the chat.
The chat from Alejandro.
Yes. So the question is, Porites colonies seem to be Pelobata, which is a cryptic species complex. Could it be that genotype and resistance to vermetids reflect differences between cryptic species? And yes, it can definitely be that. I'm being cautious about calling it a bit cautious and not saying cryptic species, because I think just looking at one region made me and not doing much more than the tree that I showed you, and not comparing it to, even though the Porites species complex phylogenetic trees are difficult, I think is maybe a way to describe it. I think that's where this is going, is that these are more likely cryptic species of Porites. I mean, they're definitely cryptic species of Porites. It's just, I don't know which one.
Okay, any more questions? I think now we are done then. More questions?
I do have a question.
Okay,
and we just tell the audience, people who are not familiar with the campus colloquia, we tend to open it just for informal discussion after the talk. So feel free to just stay and ask informal questions or whatever you want. So in terms of your future research, Anya, so you have the seagrass research that is in the Tomales Bay, so that's relatively local. The coral research, are you going to be able to do something here in California, or do you need to travel for that?
For that, I'll need to travel. So in the December, I just actually got back from a short field trip with one of my Eve colleagues to the Cook Islands, where we visited Rarotonga, which is the largest of the Cook Islands. And the Cook Islands are a country that are south of Tahiti and French Polynesia, and somewhat affiliated with New Zealand. And they are they have quite a few of these massive Porites colonies, and they have colonies with and without vermetids. And so I have some follow-up work now for this work to see just how widespread this pattern might be that I've discovered here. And there's the potential, I think, for some follow-up work to test some of the hypotheses that I've suggested in terms of community phenotypes. So one thing that happened during the time I was doing, right after actually that I finished this experiment that I described, the vermetid gastropods experienced a mass die-off around French Polynesia, and so it would be difficult to do specific follow-up work on this study in Moorea. And so Moorea or Rarotonga are two potential areas to do some tropical work. And then for the temperate coral work, that'll be lab-based. And so I'll be able to do that either at Bodega in small aquaria, or with some collaborators on the East Coast in Rhode Island.
Thank you. And the coral work in French Polynesia, do you find that same species anywhere else in the world? And if so, is there interest in going to another geographical location that has different conditions, temperature and things like that?
Yeah, no, I mean, definitely. So the massive Porites species complex is complex and pretty widespread across the South Pacific. It's a pretty hardy coral. And in fact, like in the Cook Islands, I didn't know what to expect precisely when I got there, but it was the dominant coral on the reef. And I normally, if I'm visiting a reef in the South Pacific, I do expect to see that type of or that species complex. And so yes, I would be thrilled to go to other places to think about this question.
Yeah, so as experimental scientists, we're used to experiments taking a very, very long time. But of course, you cannot transport your lab to French Polynesia for months at a time. So how does it work for someone like you?
Oh, yeah. Well, so when I was as a graduate student, I would live in French Polynesia for 3 months at a time. But I would also set up experiments and then leave and then go back and hope that it was still there. And that's a really, I think, typical way of doing that. So one of my hopes is that I'll have students that'll do some work in tropical environments and temperate environments, but potentially also hire some people in like the local tropical environments to help maintain experiments.
Yeah, so those are really interesting budgets in the grants.
Yeah, I mean, I think that there are different types of grants. Like I think that the coral reef it's two different two different types of grants to write. But yeah, it's one of the for example, like benefits of working in a tropical environment or in the South Pacific in Moorea is that the Gump station is a UC system station, and so there's some relief in terms of stay. I know who is I think Lydia at CIESE, maybe we're going to talk at some point too. That's one of the things I'm trying to figure out is where some of the coral work will happen. And
sure, we'd love to.
Yeah,
we'd love to. I'll send you an email.
That'd be great. And I think that the nice thing too is the work. I mean, the benefit of being at the marine station is that there's always going to be questions to ask right outside our front door. And the nice thing too is there's some, I think, like really nice parallels between the seagrass system and the tropical systems. And so I think there's a lot of different avenues for funding because there'll be different systems.
Excellent, thank you.
Okay, do you have more questions? Anybody else? Okay, look like now we are done. You have a lot of very interesting questions, so this was great. I love your talk and I learned a lot. So thank you so much, and thank you everybody for being here today, and see you in the next campus talk.